Hi, this is a video about sickle cell disease. Sickle cell disease is caused by a single point mutation in hemoglobin that ultimately results in generation of deformed and adherent erythrocytes. These sickle-shaped red blood cells display two important features, that is structural instability and a tendency to aggregate. Now, this accounts for the major clinical consequences of sickle cell disease, such as hemolysis and vasoocclusion. Sickle cell disease is caused by a single nucleotide point mutation in the beta-globin chain, which converts the sixth amino acid in this molecule from a glutamate to a valine residue. The disease is only seen when both alleles of the beta-globin gene are affected. Consequently, only those homozygous for mutation are affected, and transmission occurs in an autosomal recessive pattern. Heterozygous individuals are largely asymptomatic and act as carriers. Sickle cell disease is largely seen in those of African descent. The high prevalence of sickle trait in populations of African descent is thought to be of evolutionary origin, as heterozygotes appear to be more resistant to malaria. When two mutated beta-globin chains are incorporated into the alpha-2 beta-2 hemoglobin tetramer, the result is termed hemoglobin S, or HbS. Now, hemoglobin S tetramers have a tendency to polymerize under conditions of low oxygen tension, which affects the membrane properties and shape of the erythrocytes. These changes render erythrocytes sticky and deform their shape from their normal pliable biconvex shape to that of a rigid crescent. This is reminiscent of a farmer's sickle. Although these changes are initially reversible, with repeated exposure to low oxygen environments, erythrocytes become permanently deformed and sticky, yielding the two major pathogenic sequelae being the hemolysis and microvascular vasoocclusion. Rigid sickled erythrocytes are more readily recognized and eliminated by the cells of the reticuloendothelial system, such as the microglia cells of the brain, macrophages of the lymph nodes, pulmonary alveolar macrophages, liver Kupffer cells, glomerular mesangial cells of the kidneys, macrophages of the spleen, etc. So, for example, within the spleen, the sickle cells are recognized by macrophages within the splenic cord and are destroyed. This causes high rates of extravascular hemolysis. Additionally, sickled cells are relatively fragile and can also undergo intravascular hemolysis. Consequently, patients with sickle cell disease tend to display a high background of chronic hemolysis and a hypercellular bone marrow that generates large numbers of reticulocytes in an effort to compensate for the chronic peripheral red blood cell destruction. The membrane of sickle cells appear to express increased levels of adhesion molecules and is thus abnormally sticky. This results in slowing and ultimately clogging of the microvascular beds, yielding microinfarctions throughout multiple organs. Sadly, these vasoocclusive events tend to reduce local oxygen tension, further promoting hypoxic sickling of the erythrocytes, and that gives rise to a vicious cycle. So as we saw in the beginning, hemolysis and vasoocclusion are responsible for the clinical sequelae or the clinical consequences of sickle cell disease. While chronic hemolysis yields a chronic hemolytic anemia, vasoocclusion can affect virtually any organ and often occurs in acute, repeated episodes throughout life. Patients with sickle cell disease display a chronic background of both intra and extravascular hemolysis. As a result, patients are chronically anemic with low baseline hemoglobin concentrations and often require repeated transfusions. In compensation for this peripheral destruction, a patient's bone marrow is hypercellular, releasing vast amounts of reticulocytes, seen peripherally as reticulocytosis. Occasionally, sickle cell patients will have an exacerbation of their baseline anemia with an inappropriately low level of reticulocytes, indicating a lack of bone marrow compensation. This usually occurs in the context of viral infections, especially with parvovirus B19, which can suppress hematopoiesis. Acute chest syndrome is a unique clinical entity specific to patients with sickle cell disease that manifests as a syndrome of acute onset chest pain in the context of symptomology indicative of pulmonary dysfunction, including hypoxemia, tachypnea, cough, and so on. Acute chest syndrome is thought to be caused by sickling and vasoocclusion within the pulmonary vasculature, a particularly dangerous scenario as the consequently reduced systemic oxygen tension can promote massive sickling throughout the body. The syndrome itself may have a number of triggers, especially pulmonary infections and fat emboli due to bone infarctions. The lives of patients with sickle cell disease are frequently marked by intense episodes of acute onset bone pain, thought to be caused by vasoocclusion within the bone marrow. In some cases, this can lead to bone marrow infarction and the creation of fat emboli, which may travel to the lungs and secondarily trigger an episode of acute chest syndrome. Sickling and vasoocclusion within the penile venous outflow tract can lead to priapism and ultimately lead to permanent impotence in many male patients with sickle cell disease. Sickling and vasoocclusion within the spleen, leading to accumulated infarctions, is so common in sickle cell disease that virtually all patients exit their childhood with a small fibrotic spleen with minimal functionality. As a consequence, adults with sickle cell disease display an infection susceptibility to encapsulated bacteria, especially Salmonella. Thanks for watching. Please like this video and subscribe to the channel. More videos coming up. See you in the next one.